Hello, everyone. Before we get started, just a few little requests. My name is Anjali Arundekar, and along with Darda Newman and Mayanti Fernando, it is my pleasure to welcome you to our ongoing series on Dissent and to our first event of 2022. Before I begin the proceedings, Nelson Hutchinson, who is our behind the scenes mastermind, will be muting all of you for now, but could I request that you show your lovely faces if you feel up to it, because we have comrades from across multiple time zones. And one of the reasons we chose not to make this a webinar is so that we could have a feeling of intimacy and so that Bishnu can enjoy the warmth of your faces across time zones. I am currently in Mumbai, in Omni-ridden Mumbai. So a belated Sal Mubarak to you. Good evening to my friends who are in my time zone. And of course, good morning to all of you who are joining us from the East and the West Coast. Now, before I begin, I want to acknowledge that the land on which UC Santa Cruz stands is the unceded territory of the Awaswa-speaking UP tribe. The Amamutsan tribal band, composed of the descendants of indigenous people, is today working hard to restore traditional stewardship practices on these lands and heal from historical trauma. Now, I also want to remind us, as my queer friend and indigenous scholar Joseph Pierce routinely reminds me, that an acknowledgement to land is not the same as a relationship. Land does not require that you confirm it exists or that it has been stolen, rather that you reciprocate the care it has given you. Now, our Center for South Asian Studies, and many of you have shared events with us now, is in its second year, and it focuses explicitly on justice, broadly defined economic, feminist, aesthetic, environmental, social, and so much more within South Asia and the broader Indian Ocean world. Now, for the past 2 years, we have been very fortunate to curate conversations that both foreground and challenge continued histories of occupation, authoritarianism, as we have seen and continue to see in Indian-occupied Kashmir, and the foundational disposability of minoritized bodies, as we have seen most recently in the ongoing brutalities in Tripura. At the heart of our struggles remains the enduring question of subaltern life worlds within which our collectivities survive, resist, refuse, and sometimes surrender. Now, today's event, as has been our custom, we have dedicated each of our events to either a political prisoner or someone who is working on questions of justice. So today's event is dedicated to the fierce work of Meena Kotwal, a Dalit journalist and the founder of Mook Nayak, a Hindi media platform whose reportage on caste and gender atrocities has been the exemplar for courageous journalism. On December 25th, 2021, a video Kotwal tweeted went viral. In this video, she is seen burning, the Manusmriti, seen, as many of you know, by many upper caste Hindus as the legal guidebook, the Dharm Shastra of Hinduism. In an incredible gesture of protest, she writes beneath the burning of the video, "Aise hi is ghatiya kitab ko jalaenge, और ये हम लगातार करते रहेंगे, सिर्फ जलाएंगे ही नहीं बल्कि इसके विचारों को निकला देंगे, बाबा साहिब ने भी यही किया था." Here is how we burn this despicable text, and this is what we will keep doing. And not only will we burn it, but we will uproot the very thoughts behind it. Baba Sahib had done the same thing. She's, of course, alluding to a similar action Baba Sahib Ambedkar took many years ago. Now, Kotwal does not just enter, or rather end, her gesture of dissent here. With a final flourish in the video, she adds, "Aag aise lagai, maza aa gaya." I have lit the fire in such a way that it has filled me with enjoyment. Now, Kotwal, as many of you may have guessed, has received threats of death and sexual violence from many of her detractors who seek to silence her, just as they did in the tragic case of another fierce journalist, Gauri Lankesh. Now, I would also remind you that despite the virality of Kotwal's video and the threats she has received, the Indian media has been very quiet and refused to support Kotwal publicly. Caste hegemony, after all, requires Dalit bodies, especially Dalit female bodies, to be confined to the margins of speech. Now, to surrender to that narrative dispossession would be to do disservice to Kotwal's Andolan, to the gauntlet she has thrown down for us by holding out what a Dalit feminist in describing Kotwal's actions described as, quote, a middle finger to Brahminical despotism. Instead, today, in the spirit of our series and our speakers' talk, I want to stay with the maza, the visual enjoyment she emphasizes in her posted video of the burning of the Manusmriti. I urge you to watch the video, to sit with its mediated protest, and to agitate, educate, and organize. Burn the book, burn the paper, and burn the ideas that sustain them. That is where the maza lies, the joy of dissent, a joy that comes from a fire that has been lit to spread, to incite, a visuality otherwise unimaginable in a world inhospitable to the live scenes of anti-caste protest. As such, I am delighted that we have with us today Professor Bishnupriya Ghosh, whose multi-sited, multidisciplinary, and wide-ranging research sutures difficult and dense visualities of dissent and virality across histories of global media cultures, epidemics, geopolitics, and so much more. And I know that I have learned an enormous amount from her work, as of many of you who are here today. Professor Ghosh teaches at UC Santa Cruz, UC Santa Barbara. I wish she was at Santa Cruz. It's wishful thinking, and is the author of 2 field-changing monographs: When Borne Across, a literary cosmopolitics in the contemporary Indian novel in 2004, and Global Icons, Apertures to the Popular, 2011, which I find to be a very queer and exciting title. She has also co-edited the Routledge Companion to Media and Risk, and has a brilliant new monograph, The Virus Touch, Theorizing Epidemic Media, which should be forthcoming from Duke University Press in 2023. Now, before I have the pleasure of turning it over to Vishnu, just a couple of rules for our Zoom protocol. Number 1 is Nelson will spotlight the speaker during the talk, so that you'll have the pleasure of seeing her. Please, you'll be muted, but we love to see your faces. If you have any questions that you'd like to pose, please message me in chat privately, and I will put you in a queue, and I will invite you to ask the question yourself. Of course, if you don't want to, I'm happy to do so, but that's worked fairly successfully for us. We are also always hammered by trolls, but we have a very dogged team working behind the scenes to make sure we are not blasted with unfortunate images, and hopefully everything will go well. So without further ado, let me turn it over to Professor Bishnupriya Ghosh.
Thank you, Anjali, for that introduction, and for actually inviting me to this amazing series that I've followed quite closely. And when you invited me, I was thinking, how would this part of my work, unlike Global Icons and the work I've done before, fit your center's theme of dissent? Because the dissent here is, of course, not readily palpable or readily legible, but we'll get to that in a bit. I wanted to mention that beautiful image of a mask lying in Elysian Park in Los Angeles is the work of the artist Pato Herbert, who's interesting. I actually don't get to that image. It kind of fell out of this chapter and I deal with it somewhere else. But I do want to mention his work. He has now taken these photographs. He's a COVID long hauler, one of the earliest people, earliest instances of COVID that I know. And so he's been working on the COVID HIV AIDS relationship. And that's why I use that image. He's now got this series up. It's called Lingering. And he has it up on silk screens. And I believe it's going to show at Pitzer College. So, you know, it's an up and coming thing. And I'm hoping I'll deal more substantially with him in the project that is now just about falling out of this one. But I'm interested in the conversation. Thank you, Nelson, for all your help with you know, getting me on track with images and for disseminating this talk. And what I'll do is now share my screen. The talk turned out to be more about monkeys than I thought it would be, but we'll get to get there in a minute. Okay, so one last thing about media here. Some of you may have also recognized the song, you know, Nena Cherry's song from Red Hot and Blue, which was an album for AIDS benefit that came out in 1990s, right? So, and it will make sense as the talk goes on. Okay, so let me get to this question of distribution in this mouthful of a title, by starting with the Human Microbiome Project. We are not alone. Once that iconic observation compelled fantasies of alien invasions and red planets. Now it discloses microbes that make up the human. If the Human Microbiome Project, or the HMP, tells us that microbial cells outnumber human cells 10 to 1, scholars anoint this distributed human, Homo microbis. Now, this illumination of this ever-swarming bio-body, which is sort of the basic start of this talk on distribution, also establishes microbes as significantly beneficial allies in ensuring human health. Against this ecological tide, though, alarm at microbial abundance surfaces iteratively in health emergencies like this one, a globally synchronous crisis event of a pandemic. Among these microbes, pathogenic viruses, as we know, have commanded a lion's share of attention in every flu season and every viral outbreak, recasting microbial human relations as warlike. And I'll just play this video in the background. I won't really talk about it, and let me put off the sound. This is a public health message that was made by a Stanford nonprofit called Teach AIDS in the Bay Area. And what's interesting about it, it's a modular piece of software in which you can kind of plug in your own country wherever necessary to talk about the immune system's response to viruses. Okay. Like many public health messages, it's in cartoon form. So I'll just play this in the background while I talk. Now, this warlike stance that you'll see here is what is at stake in rethinking health in the more than human Anthropocene. And I'm using that term with all the baggage that comes with it. Against the dawning realization that we humans have lost the war on germs comes a perception of health as a moving target whose goals lie beyond medical advances. HIV is now called a hyper-endemic because 60% of HIV infected are on antiretroviral therapies, while others still struggle for access. And SARS-CoV-2 is tending towards endemicity. Clearly, we don't seem to be winning this war on germs, even as human activities continue to generate conditions for future emerging infectious disease events. And this is what I'll focus on. Instead of pitting humans against viruses then, epidemics call for a well-crafted multi-species politics that continue to decenter the human while addressing existing and potential losses within species. And of course, many of us in this pandemic are coming out with grievous losses that we'll have to deal with for decades. So this is the effort is to think both together. Unthinking anthropocentrism, such an orientation emplaces precarious life in an increasingly precarious environment. The Virus Touch, the book that I've been working on for the last 10 years and really needs to be finished, is devoted to how the extreme situation of a global pandemic compels a recalibration of multi-species politics. Experienced as fever and fret in individual bodies and across them as infection, an imminent multi-species relation confronts us during the epidemic. Currently, this is the SARS-CoV-2 and human relation. Such a novel relation appears to us in image, milieu, number, and movement. For instance, your PCR test captures the viral load as a numeric distribution, X amount of virus in Y amount of body fluid. Indeed, we have learned a great deal about PCR spins and cycles that transform multi-species relationalities into mathematical distributions of viral particles in vital media as the baseline for clinical interventions. Now, these technical mediations make multi-species relations intelligible by enacting what I call epistemic cuts in dynamic, changing multi-species assemblies. There is mastery in these mediatic processes in the extraction and objectification of one multi-species relation as discrete, isolable, and epistemic object plucked from its environment. But ongoing challenges to that mastery ultimately activate another kind of knowledge altogether, sense perceptions of our irrevocable entanglement in living systems that activate making connections between domains of specialized expertise. And this is where the Dissent will come in. The question that animates the book is, what can epidemic media as a research concept offer pandemic futurities? Now, this book began many, many moons ago with the last great pandemic in recent history, the long wave HIV pandemic that closed out the 20th century. Writing during the present COVID-19 crisis that stilled all life as we knew it in March 2020, I found my generational experience of HIV/AIDS suddenly historical in the flash of a spillover event. As the COVID-19 cataclysm began to transform all frames of reference for thinking about epidemic media, and it has been a hard book to finish, the HIV/AIDS pandemic remained a touchstone because of what its formidable knowledge domain has set in motion. And this is the current epidemic episteme I'll return to in a minute. In keeping with the center's theme of dissent today, I focus on what I see to be, in what I see to be critical discordances between knowledge practices that constitute the range of epidemic media that I assemble in the book. And I just put this up because really I'll be talking here on chapter 1. But the chapter 3, 4, and 5 are more substantially about epidemic media, and they talk about the image, medium, and movement as mediatic categories which actually move across domains of study, even within media studies. Okay, so what do I mean by an epidemic episteme? I understand an episteme to be a historical configuration of knowledge that defines the conditions of possibility for all other knowledge practices, a kind of paradigm shift, as it were. The paradigm shift in question here is a substantial change in thinking infectious disease emergences in the late 20th century. First defined in the order of things in 1966, in subsequent writings, Foucault suggested several epistemes may exist in a historical time. His notion of episteme is most often compared to Thomas Kuhn's much discussed notion of the paradigm in the Structure of Scientific Revolutions in 1962. But where the paradigm is a conscious disciplinary matrix, epistemes are orderly structures, Foucault argues, that underlie scientific knowledge, but that might remain invisible. Now, the historical paradigm shift activated by the emerging infectious disease emergences in the 1980s reverberated across the human sciences, for sure, but it also settled in wider public consciousness, partly spearheaded by HIV-affected communities that routinely engaged with new scientific findings and existing protocols. And this is a very well documented history. Amid debates over clinical shibboleths, medical affordances, and healthcare provision, Cindy Patton, Paula Trickler, and Stephen Epstein unerringly demonstrated how science was indeed a cultural complex. As epidemic panics mounted and the death toll rose, scientific enclaves and larger public cultures were rife with anxieties and fears, desires, and prohibitions, caught in what David Abrams, the anthropologist, has called a flux of participation that obviates neutrality. For all these reasons, one might say that the 20th century paradigm shift in conceptualizing infectious disease emergence is an episteme. Now, one of the most notable features of this episteme that I situate in the HIV/AIDS pandemic knowledge domain was the ecological take on health. HIV emergence, along with Marburg, Hanta, Ebola, and to some extent Lassa in the early 80s, compelled an orientation towards microbial life from planetary recesses, crossing into new host populations. As scientists gathered around the emerging infectious disease events and their zoonotic agents, in 1989, Joshua Lederberg, the scientist who coined the term microbiome, christened pathogenic viruses as the deadliest threat to mankind. With the introduction of a new course on the infectious disease at the CDC, we passed into what Nathan Wolfe termed the age of viral storms, and we have actually been there ever since. The burgeoning literature on animal reservoirs told us that viruses live in opportunistic tolerance in some hosts, causing little trouble until changing conditions create new conditions for viral mutations and cross-host transmission. Now, in media and cultural studies, the emphasis on species, the sharing of biological resources, that is parasitism, and ecological ones, habitats, food, water, and I'll get to the story a bit later, made landfall in what is called multi-species studies. Implicit to this field was a critique of anthropocentrism, underlying readings of pandemics as global public health crises. So the other part of the story is public health experts who've struggled with HIV community transmissions and its morbidities. Responsible for 33 million deaths from AIDS and 38 and more million living with HIV AIDS worldwide, discontinuous HIV/AIDS epidemics soon became a global pandemic until the 1995 retroviral therapies turned the tide. One could read the subsequent story. Usually we divide the period of early AIDS and the post retroviral story as a kind of a periodic disjuncture. The subsequent story of what is now known as managed HIV is a tale of biomedical triumphalism. Yet the critique of global public health as a biocapitalist enterprise made visible persistent geopolitical imbalances rooted in histories of colonialism and racial capitalism that sorted segregated host populations and distributed health across the world. And there were so many, of course, as we know, pharma fights that came out of this recognition. The pursuit of infections and inequalities, as Paul Farmer has characterized them, drew attention to the differential vulnerabilities that challenged any unitary anthropos in species discourse. In such diagnosis, it was not the zoonotic spillover that was the problem, but long-term health inequities among other social systemic failures that transformed that spillover into a cascading disaster. And there, I don't really need to trot out any COVID-19 stories to make the point. On the media and cultural studies fronts, the study of individual and collective illness and dying materialized across interdisciplinary domains, such as the medical humanities and the social studies of medicine, and many others, right? I'm just mentioning a few to talk about these sort of different disciplinary domains that appear in the book. Now, today I turn to two stories from South Asia representative of divergent analytic trajectories rising in the current epidemic episteme. One follows the global health crisis, the other, the spillover event. One is preoccupied with bodies, medicines, housing, food security, and all that makes health. The other with deforestation, illegal wildlife trade, industrial farming, and all that makes zoonotic spillovers. And these are, of course, 60% of EID events. Dissent, then, is not a disagreement about what health is or what it should be. It is not even about different objects, different scales, temporalities, and agencies, questions, and terms. Rather, I would argue it's a disciplinary disjuncture whose critical discordance is crucial to rethinking what health could be for pandemic futurities, were we disinclined to repeat history as farce. So let me go into the South Asia story, the first story. This is Urmi Yadav, they were one of my interviewees in 2018. And this is the Humsafar Trust's drop-in center that actually came about after my interviews. As India's largest NGO promoting LGBTQ rights, Humsafar has had counseling and healthcare advocacy as part of its folio since 1995. When I conducted the interviews, Sanjeevani was the registered community-based organization. It takes its name from a hospital chain in Mumbai, and it served the men who have sex with men and transgender communities, and spearheaded the HIV AIDS treatment since 2010. Very briefly, at the time, the Sanjeevani team was the institutional interface between people living with AIDS, 10 government art centers, and 5 hospitals across Mumbai. So here on the left, you see the Sanjeevani as it's nested within this larger, the Humsafar facilities. The facilitators handle registration and temporal records of individuals under their care and streamline testing and treatments for HIV co-infections in several hospitals, interfacing with global NGOs such as the Médecins Sans Frontières, MSF, for the second and third line multi-drug resistant treatments. Sanjeevani focuses on ARV adherence and therein the maintenance of the viral threshold. As such, it is part of India's VHAN, or a loss to follow program, that follows up with patients who have fallen off their meds. And therefore, it's a part of a worldwide biomedical agenda. Now, at the time of my visit, and for many reasons, I've drawn the photographs from publicly available ones. At the time of my visit, the Humsafar offices offered physical facilities for storing medical files and the digital infrastructure for tracking adherence. Each PLHIV, or the friends as they're called, had a file in the form of a ruled exercise book that was populated with hand-transcribed data from tests every 6 months. The classificatory system for these blood files registered the somewhat precarious circumstances of the friends. Since strong social stigma prevented some from keeping records at home, their medical files were stored at the Humsafar Trust office that you see here and filed under the friend's mother's name. This is a nomenclature that protected against disclosure. So following Cornelia Wiesmann on files as an accumulative storage technology, I characterize these records as the blood files because the periodic blood tests of the viral load test in particular organizes the data entries. The files accumulate standardized clinical data as well as informal notes on missed visits, new co-infections, and other vital statistics. The informal notes embed the numeric blood composition within the patient's disease milieu, as epidemiologists would call it, and singular life circumstances. In accordance with well-established protocols of translational medicine, the Sanjeevani mesoscale health experts translate technical notations for the friends, documenting everything from urban conditions, their neighborhood changes, housing changes, transportation routes, new medical facilities, but also contingent events like weather disruptions, government policies, and so on and so forth. Now, if we look closely at the blood files as epidemic media, 2 modalities of distribution are immediately apparent. In the biotechnical inscription, of blood substances like HIV-1 RNA, and this is the one I've been tracking, you have an inscription that has a numeric representation. The viral count is privileged for sounding out the patient's health. Staying undetectable, as we know, is to keep the count below 50 copies of HIV per milliliter blood. A threshold for the viral load test, as I have discussed elsewhere, has evolved over time into a gold standard. So this was also a part of a longer debate. Now, within global HIV/AIDS global medical infrastructures, as clinical translations streamline test outcomes from bench to bedside, we find a predictable distribution of value in the knowledge economy. Creative labor and technical expertise appears to flow from the R&D enterprises of the global north to capillary points of care in the global south. Small-scale outfits accrue value largely as beneficiaries of global public health dispensations. This distributive politics, I will not belabor because it's been amply analyzed by scholars, Kaushik Sundarajan, Melinda Cooper, Catherine Walby. I mean, there are an N number of people who've talked about this. My point, though, is that it informs the structure of my book, so that a range of healthcare organizations, I include a range of different kinds of healthcare organizations in thinking about epidemic media, as an attunement to the massive distributed creative labor that made HIV/AIDS a hyper-endemic. So this is sort of what has come out of the formidable history of HIV/AIDS activism. I forgot to show you this. This is not, I'm not really looking closely at this. What it is one of the epidemic sort of fundamental or foundational epidemic images, which is the curve. And there's been a lot of writing about how the curve, as it changes with the feedback of both infection at the populational and individual level, is a crucial epidemic image. Now, the viral load test organizes the chronology of the blood file. So we might ask, how does it mediate dynamic multi-species assemblies? Clearly, what is at stake is an unsustainable biological sharing of vital resources. But this is not how the test represents the matter. In the numeric representation, viral particles emerge as value-bearing objects against a quiet, volumetric backdrop of blood. Such objectification relies on epistemic cuts into living systems and processes, the extraction of individual blood specimens, apportioning each into multiple samples, and then preparing them with buffers and so forth for molecular analysis. And this is sort of the close lab work analysis that I do in part of this chapter. The study design or protocol that accompanies each specimen shipment determines the constitutive elements, whether you're looking for T cells or HIV-1 RNA, these constitutive elements, and calculates the sample space, that is X millimeters of blood, according to these elements, and therein generates a set of probable outcomes. What does not fit numeric parameters often registers as error, noise, or redundancy. And here I am influenced by my colleague Greg Siegel's work on noise, the noise in black boxes when you see them from plane crashes, and what the noise, how productive it is to look at the noise. Now, the procedural ambit of the viral load test was most apparent when one of my interviewees at the lab made an offhand remark about blood specimen processing on the morning of my visit to the laboratory medicine facility, and this is at the University of Washington. They had to process the sample several times because the viral load was off the charts. It did not fit plotted probable outcomes. That virus, they said, was so full of, that blood, they said, sorry, was so full of virus, it could have walked on its own. Here for a moment, the organismic configuration of the virus as a different kind of non-human agent trumped the molecular viral particle. But on most other occasions, whenever I brought up the species questions, this was anathema. It was simply not what was important in this domain of specialization. In this remark, the virus had acquired the fuzziness that Hans von Reinberger writes in the case of the gene, as it was reconstituted at the edge of specialized scientific practices. Pointing to the historical construction of such epistemic objects, viruses and genes, science and technology studies scholars have long highlighted the material constraints, including media technologies, cultural techniques, and study designs that shape these objects so that they appear as such. For the viral load test, as we know, PCR machines can drill down only so far and no further, hence the states of undetectability. This material constraint may recede over time, and there's ongoing work on PCR machines, of course, but what it exposes is the limits of technical mastery over lively materialities. Now, collaborative research in the sciences, of course, recognizes the limits of specialized domains, making all kinds of partial connections, as Isabel Stengers puts it, between differing visions of the world. But if pandemics are biological, social, and ecological emergences, complex events, the point is to think beyond the life sciences to make tactical compromises on common targets. What might it mean then to think of the primacy of the viral load test as it defines health, the premier target of pandemic intervention? But this is precisely what social science medicine has done with HIV since we have the 4 decades of research. An emphasis on viral counts, they argue, tend to divert attention from other clinical signatures such as lipid dysfunctions that might spur non-adherence, as well as from public health systems that make living with HIV possible. Health here is less a numeric threshold and more the outcome of social relations and processes. If we return now to the blood files, we find them to be baggy accumulations that cross-hatch personal patient reports with marginal notes and tables of blood data. As Sanjeevani counselors struggle to reconcile singular patient needs with the macro imperatives of viral load tests, they make notation of other elements defining the disease milieu: weather conditions, nutritional change, water scarcity. No longer numeric distribution, the blood files underscore the socioeconomic distributions that make health a deliverable public good. Now, this is a relatively simple illustration, and many scholars, of course, move across the aisle and do both. But we can see how the same epidemic media, the blood, can point researchers in very different directions. Each chapter in the book performs these partial connections between material settings. In this chapter on clinical translation, I employ a media category, vital media, to track blood across research laboratories and point of care clinics. Staying with the critical discordance, discrete epistemic objects appear and disassemble into living processes and relations. As I turn to the second story, in which this disjuncture is even more, epistemic objects become fuzzier still in the emergence of health as a multiform horizon in the current epidemic episteme. So let's move to then the monkeys here. I forgot to show you this. So, the way in which blood is treated has a long history, and there are many ways, many kinds of epidemic media, which I look fairly closely. This is the optical, and that's the computational. And the one on the right is particularly relevant for thinking about viral strains. And we're, of course, in the age of viral strains. Okay, but let me move to the rhesus max in Bangladesh. Now, this story begins with species taxa that immediately raises thorny questions, and I have had many over the course of my presentations, conceived along classificatory logics similar to racial taxonomies. Crafting species difference imposes an impossible organismic purity. Stephen Hanreich notes, organismic difference is especially difficult for microbial integrities, as we know from histories of their coevolution and rhizomatic assemblies. This implosion of organismic difference is implicit in the very constitution of animal hosts as multi-species. They are always already not one species. Yet epidemic media on animal host movements, which is the subject of the 4th chapter, culturally perform them as isolable targets of intervention, and necessarily so. Among animal hosts, non-human primates, or NHPs, rodents and bats, are premier reservoir hosts for periodic zoonotic spillovers. In recent years, the rhesus max concentrations in South and Southeast Asia have attracted a great deal of attention. Researchers flag their changing movement patterns, as well as mutating viral strains that can potentially become pathogenic. The temporality here is another kind of distribution. It's anticipatory, the statistical modeling of probable occurrences of sudden pathogenicity into the not too distant pandemic futures. And here you can see the work on risk media actually folding into this part of the analysis. Now, the second image is a documentary image from a study of the rhesus macaque human interactions in the Borne region of Bangladesh. The NHPs are epistemic objects, doubling as mediums for viruses that hitch a ride on them. Equally critical is a third configuration, an organism environment assemblage, as Jennifer Gabrys characterizes it, which is the locus of environmental study. Now, part of the study design samples habitats that humans and NHPs share, and these are what are called human-altered environments, both urban and forested. 3 modalities of distribution work together in this complex picture. The identification of viral strains in animal hosts, I'll return to this, the changing distribution of NHP and human populations within an area, densities inextricable from social and economic apportioning of resources, and the spatial distribution of planetary resources severely impacted by anthropogenic drivers. As environmental media and animal studies scholars maintain, deforestation in particular is a prime anthropogenic driver of zoonotic spillovers, radically changing habitats, nesting niches, water sources, and food, and therein dynamic multi-species assemblies. And this sort of intuition sort of sits very uncomfortably when you're in the middle of a large health emergency of the kind we are in. In this case, receding food, fruit, and water sources have created more physical contact and conflictual situations between human NHP populations, including bites and scratches. The environmental agenda is not the colonial practice of conservation, but an effort to find pragmatic solutions to potential conditions for pathogenicity. Scholars of spillovers, of zones of virulence, of feral ecologies, and this, many of you may know the collaborative project, Feral Ecologies, which Anna Singh is a part of this. They've variously argued for thinking natural and social entanglements together, since there is an overwhelming evidence of ecologically unsound practices, extractive mining to illegal wildlife trading, that reconfigures multi-species assemblies, catapulting what was once beneficial mutualism or just absent multi-species relations into difficult co-presence. Zones of virulence, as epidemic media, take spatial form, varying widely from cartographic schematics to geopolitical satellite images of animal movements, and they articulate viral, animal, and biodiversity distributions together. Now, to show you where this is, you can look on the left. There are 6 areas in Bangladesh, 2 forested and 4 urban, that are being tracked for the changing viral strains in these monkeys. The study I'm drawing on was conducted between the University of Washington's National Primate Center and the Jahangirnagar University in Dhaka. With 3 retroviruses have been studied here. And one most studies is the Simian foamy virus, or the SFV, which actually now doesn't pose a pathogenic threat to cats, cows, or humans in its present configuration. But it is closely studied for possible recombinations of viral strains. If you look on the map on the right, you can see how the strains, particularly the Borme strain, right? The strains are in different colors. There's a kind of a cluster, sorry, what am I doing? Yeah. there's a cluster of strains. You have the other strains also make it into the Borme region. So as the forest recedes, you have more and more NHP populations carrying the different strains come into a particular area. And I'll talk about this in a minute. So why is this a source of concern at all? Well, once more here, HIV and its knowledge domain gives us an answer, that it initiates this molecular scale genetic tracking within and across NHP populations. We know that the Simian immunodeficiency virus, or SIV, was tolerated by wild primates, and it became pathogenic in its zoonotic spillover to humans as HIV-1 and HIV-2. What is important in this story is less the phylogenetic evolutionary trajectory, and that's what those branching maps show, but the possibility of co-infection, a horizontal effect that motivates viral strains to mutate. First detected in 1990, we know that two strains of SIV-CPZ, which became HIV-1, combined, mingling in a chimpanzee co-infected with both. Here, horizontal gene transfer stemming from co-infection enables viral evolution, displacing the logic of Dissent. The jury is still out on tracking the original reservoir for SIV strains. And this is, it's been a holy grace and also, I think, a red herring. But what is clear is mammalian sympatry, or the overlap of phylogenetically proximate animals in a geographic area, is a factor in zoonotic shifts. We might remember that the genetic makeup of simians is 93.5% similar to humans. Now, viruses, if you were a virus, would search for similar hosts that can tolerate their parasitic needs. In other words, genetically similar mammals present already familiar internal environments for parasitic survival. As the forest recedes, then, as different NHP populations concentrate in fewer sustainable habitats, the threat of co-infection increases. So you see here, this is sort of the classic epidemic media that I look at. This is, you can see on top, you have the genetic strains, and then they are visualized in color. And then already here, you can see the Borme strain, which is red, actually moving into the other areas. And you have below the blue little dots as showing the recombinations. As we see here, the epidemic media in Bangladesh rhesus mark studies map viral strains according to geographic regions. The microsatellite DNA map made from blood draws of 126 marks from 6 sites in these studies uses the region as a functional shorthand for the viral strain, much like we use Delta Omicron here for SARS-CoV-2. But emphasis falls on the genetic code mapping in data visualizations like this, with recombinations triggering further scrutiny. Beyond gene mapping, the processes of mediation involve the capture, the darting, the sedating, the blood draws from NHPs, and the transport of blood to the National Primate Research Center for fabrication. So I'm interested not just in media representations, but also the processes of mediation that enable maps like this. Trained veterinarians, animal specialists, and wildlife biologists work together to make this media. And of course, there are many, many other kinds, and I've given you a similar one for HIV. But all this is possible because a selected habitat is sampled by what is called field specialists who collect all relevant data. This is observational, technical, so part of my chapter looks at camera traps and drones, or physical matter, blood or fecal matter, for instance. Central to this endeavor is the transect, an environmental study design that prepares the habitat for analysis. And actually, this rhesus mark study didn't make it into the book because I was tracking HIV. What did was the literature on HIV, which is does transect at the border of the DRC, the DR Congo and Uganda. And the person I've been working with who's been really kind enough to share her transects with me really taught me how to look at the sample space that is constituted by this method. The transect, as you see here, from Anne Lardisauer's diagram, is a straight line. You see two straight lines cutting through a selected part of the habitat. Researchers collect data from a measured space, say 10 meters, on both sides of the line. And in some cases, in this case, because there's an arboreal canopy, from the vertical space of the transect. The sampled habitat then serves as a basis for modeling changing distributions of population densities, of dwindling food and water resources, of soil salinity, and so forth. Global projects such as US-funded Predict1 and Predict2 base their predictive surveillance on these biodiversity data. One of my research sites for Bangladesh has been EcoHealth Alliance, a global outfit that is a part of the Predict projects. And of course, you've heard of Peter Daszak, who's been working with the Wuhan Institute of Virology. And he's, of course, there's been a lot of controversy around EcoHealth Alliance. These EcoHealth Alliance conducted a series of interviews on the rhesus mark human assemblies in Bangladesh, where interviewees in the Maradipur district noted that the government initiative providing bananas, cucumbers, and nuts for simians between 2006 and 2009 had significantly reduced simian presence in backyards and in agricultural areas. Working with collaborators, the Alliance proposed replanting of fruit trees as buffer zones between human-dense and NHP-dense habitats. And this is a kind of a locally salient, pragmatic solution to emerging environmental risk environments. So they're not really saying stop all deforestation. No country is going to go for that, right? So they come up with these solutions. Such efforts exemplify the WHO's principles of structural One Health that articulates human health with animal health and biodiversity agendas. Since the viral storms of the 21st century, such multi-sectoral alliances between molecular biologists, veterinarians, biodiversity advocates, wildlife enthusiasts, indigenous experts, local farmers, educational institutions, and policymakers have mushroomed. These exemplify what I was getting at, a tactical compromises over different visions of the world. In the book, that tactical compromise materializes in the very disjunctive stories and situations like the two you heard today. But these are critically discordant, not just because they take on different objects, but because of the very different disciplinary conversations they mobilize, even in an interdisciplinary configuration that is environmental media studies. And so there's a huge literature now on life and non-life, human and non-human. And so to sort of centralize one seems to be the failure of the other. Today I've tried to hold these disparate strains together in the pandemic logic of distribution. I've gone on too long, but let me just close very quickly with COVID-19. Epidemics, then, I'm arguing, force us to think from the point of the pathological, as Georges Canguilhem had taught us. When life surfaces at its most exigent, the immediate orientation is to find technological panacea. And of course, the antiretrovirals were of epic significance to my generation, and the vaccines are to ours. But modern epidemics have equally forced a rethinking and restructuring of health for individuals, populations, and the species. The HIV Aids pandemic showed us health was not the purview of medicine alone, that it extended to the care of life in therapies and wellness regimes, in housing and food security. And I see many folks who've been writing on this in the audience. Health could not be left to free market vicissitudes. It could be the global commons. Nor could health be constrained to human health. It had to be cross-hatched with animal health and biodiversity sustenance. 4 decades later, then, we are at another pass, having to make imperfect symbiotic agreements with what are now called awkward and flourishing creatures whose sudden abundance spells are waning. The multi-species politics that asks us to re-entangle with microbes is not to exterminate them, as we know from drug-resistant bacteria, nor an over-enthusiastic embrace, but to interrupt and dismantle known anthropogenic threats that instigate pathogenicity, and that there's a lot of work being done on this. Health as a common target concretizes the symbiotic agreement, even as every pandemic ups the ante on what is to be done. So canny multi-species politics, I'll close here, enfolds a judicious biomedical techno-fixes alongside long-term environmental solutions. Those solutions are kind of hard to think of in the middle of a pandemic. Living with endemic infection is one form of entanglement, a making together in Donna Haraway's terms. The creative process of adhering to HIV therapies has been an experiment in living artfully with technological enhancement. If re-entanglement mandates a multi-pronged strategy across domains of expertise, as I have argued, this is where critically discordant scholarship is useful. It stays with fuzziness, with error and noise, seeking conceptual lines that traverse fields of study. Such discordance, and many scholars actually work on this and bring these two sides together, such discordance performs a multi-species politics that renews the national and social contract of being with other species as well as one's own. Thank you.
Thank you so much, Bishnu, for that wonderful talk. And if I could again invite all our folks in the audience, you know, to if you feel up to sharing your videos, that would be great. So Bishnu could have a look at who's here. I know that I have a billion questions. Bishnu, your talk was, you know, full of so many provocative directions that one could go in, you know, so I'm sure there are others as well. I have, let me start while people are sort of meditating on the many offerings you've given. I had a question that was, I hate to use the word clarification because it moves away from this discordant, you know, gauntlet that you've thrown down for us. But I was trying to understand the logic of the epistemic cut in relationship to this multi-species communication. And I was thinking about the epistemic cut through the language of the sample. So for example, you started off with, well, the two exemplars you used was one of the sample, you know, in Hamsafar, and the file and the disclosure and the kind of fuzziness of that environment, and then the sample from the monkey, which is, you know, both a multi-special communication, but a very different environment for the sample. So I wanted to sort of, you know, have you say a little bit more about what happens to the signature of the sample, right? Which we know, like the inequity of the global South and the North, we know it to be flawed and its conditions of possibility always corrupted. But the sample endures, right, as a form of mediated communication, right? Whether it's through the story I began with, the exemplars we use. So I wanted you to say a little bit about, you know, what that sample is doing in your own work. And second, and this you don't have to answer, but you ended with COVID, so I have to ask, is COVID is airborne, right? In a way that's different. So it's about the kind of environment of exchange, which has also made the epistemic orientation to it slightly different. So I wanted, you know, I know that's not the purview of your book, but you are the virus touch, so we're going to have to ask you about COVID. So whatever you fancy. I know it's a lot, but, you know, whatever you want to answer. I know there are lots of other people who have questions, but let me start with that. So thank you very much.
Yeah, well, thank you for that question. I know you work on the exemplar. So, you know, we've had this conversation about what it means to take a piece, right? And look at it as an exemplary of the larger environment. Now, I think what I've been interested in the sample is the sample space. So, you know, if you take, for instance, a pack of cards, right? And you say, the elements, the constitutive elements are clubs, diamonds, hearts, etc. right? or if you switch gears and you say, well, it's going to be the king, queen, jack, whatever, right? So those are two different kinds of constitutive elements, and they make up, or they create the parameters of the sample space, right? So that's one of the ways in which I've been thinking of whatever you choose to look at, right? So in thinking about, the transect is the other big sample that I look at. It's already designated, you look at particular things and you don't look at others. And one of the things that in the blood processing lab, and this is absolutely necessary, right? I'm not saying this is a mistake. I think what science and technology scholars do is to look at the construction and what it may leave out, which then leads you to something else, right? So what is outside of the sample space? And so in the blood processing lab, for instance, whenever something else, like a co-infection, right, would skew the sample, they'd say, well, this sample can't really be used for this study, but it would be discarded, as these samples are. The samples are many of them, so I would say, but what is in that sample? And they'd say, well, there's a kind of a redundancy or an error there that is not relevant. Yeah. So the sample space. So those kinds of things were interesting to me, not simply as, the post-structuralist move of saying, oh, these studies are limited. Every study, every domain, including our own, is a limited. But I was interested in how that space is marked and bordered because of what falls out and what that leads us to. Yeah, so when I first went into the lab, they said, well, it's fantastic to have a media scholar. Do you work on public health messaging? And I'm like, no, I'm actually very interested in the PCR machine as a media technology. And they're like, it's not a media technology, So there's a complete disconnect between what is considered media, for instance, right? And the thing that we do best is public health messaging. And I started with, the public health message. Now, what that tells me is there's this kind of partitioned domains of specialization, and scientists are fantastic, I think, at working collaboratively, say on HIV. That's why we know so much about it. It's probably one of the best known living systems because of the collaboration. But there's a rare collaboration outside, and I find this among social scientists as well, that the privileging of science, right, or the techno-scientific, right? So seems to displace what's really crucial and grievous and hurtful at this time, which is the loss of human life, right? So I'm constantly caught within, and so many of environmental media scholars have had this conversation with, to look at the biological is to privilege life over non-life. And so the sample space, Anjali, is interesting to me in what the kind of conversation it sets in motion, right? And the terms and conditions under which we can talk about health. Yeah. And what I'm trying to suggest is nobody wants to hear, think of how hard we're trying to pin COVID-19 down to that Wuhan Institute. Nobody wants to hear about animal health. Right? But that's sort of a very long-term thing that we actually lose if we don't move across an ecology of practices. So that's really what I'm getting at in talking about how the sample space might delimit a second constitution of the same object, if that makes sense. Yeah.
Yes, very much so. Yeah. I have a question in chat from someone who wants to remain anonymous, is a graduate student, and I'll read the question, but I want to first also open it up to people. If you have a question or a comment in search of a question, this is a conversation. So, just message me, or if I see you, raise your hand. So Bishnu, while people are thinking about your comments, the question in chat from a graduate student is, loved your talk, learned a lot. but is there a way in which South Asia can ever be a space of discordance rather than compliance in histories of the epidemic? So I think the question here that's being asked is, yes, we know the inequity exists and we're attuned to it, but is there a way out of it, or we always surrender to that form? And I think you gestured to that in the opening remarks. So that's the question from the graduate student, who is part of a cluster we have here on science and justice.
Yeah, so within, it's a great question, actually. I think if you're talking about the global biomedical infrastructure, right, you are caught in this compliance model. And, it's very interesting that the first samples that were taken, I think from Liberia to the Pasteur Institute, the first HIV samples, Louis Montagnier actually said that we can't really, this is a kind of a knowledge economy where we're getting raw materials from Africa and expertise is being produced here. So, from the beginning of the pandemic, this kind of unevenness was there. So one of the stories, I'll say two things. One, South Asia has, the pandemic histories or epidemic histories tell us it's actually not been a place of compliance. And particularly if you look at the beginning of modern epidemiology with cholera, right? It started in South Asia from the mangroves where the cholera vibrio jumped into humans, and the British soldiers who traveled back on ships took it back to England, 1820s. This history has been written, 1830s. And then you have that map that John Snow does in London, right? And that's because of the cholera outbreak in London. Now, as we know, and David Arnold has written extensively about this, about cholera, but also smallpox vaccination, right? There were many, many other perspectives on health that were fought over at this early vaccination moment. So the disagreements around what medical panacea can deliver and how, right, is very different in different parts of the world. So my other site is South Africa, where I've been talking about these very small HIV adherence clubs where people get together every Thursday and talk about their meds, and they give blood and so on and so forth. It started out as a one woman's initiative, right? And it's really interesting, Fanuela Ganshu, and she's been working there. And there too, the conversation has always been about HIV and tuberculosis. And there's been a lot of innovation from this outfit that now the MSF has made global, right? So what we see as the global biomedical infrastructure has its own buried histories in which debates, and I don't need to even say this with HIV AIDS activism, right? The clinical trials were changed forever after that conversation, right? So there's, but it's very quickly we get into the story as I think I saw G. Fay and others, that volume on AIDS and the distribution of crisis. We know how that story goes, where it becomes a tale of biomedical triumphalism, right? So yeah, it's a complicated question. It appears compliant, and I think it's necessarily so if we are to get free therapies. But South Asia was, companies produced the generics. What was that? It was not compliance, right? And I think if we get antivirals now, the Indian companies will be making generics in a jiffy, and the global South will be much richer for it, right? So.
Yeah, I mean, I think that's a wonderful response. And I think, historically speaking as well, we know, as you reminded us, that has always been the case. My wonderful colleague, Junaid Sheikh, has a question. Junaid, take it away.
Well, thank you so much, Bishnu, for that wonderful talk. It's so lovely to see you too. I had a question about, you. In the middle of the talk somewhere, you mentioned about the massive creative labor that is involved in maintaining an epidemic media, and by extension, of course, the epidemic episteme also. And I wanted to ask you more about that labor, because in the images that you showed of the Humsafar Trust, for instance, there are people maintaining data and filing who are playing let's say, a low-level role in maintaining this media and constructing this media, an important role, but a low-level role. So I wanted to ask more about that. First, what are some of the relationships of power in maintaining this the epidemic media? And B, what happens to the data that they have collected, the Humsafar Trust, for instance, the knowledge that they have collected? It seems to me that this knowledge, this data can be easily sold to pharma companies, etc. So I wanted to know more about that too.
Thank you for that question. I don't know if I can answer the latter part. It's great to see you too. Great to see actually so many friends from the UCs here. I would say, the first part is easier. There's a book, Melinda Cooper and Catherine Wolby's, Clinical Labor, in which they say, even if you wake up every morning and take your HIV pills, that is clinical labor. So they're talking of when so many people have talked about this, Kalindivora, etc. The oocytes, and there's a biological labor at one level, right? And then, of course, there is the immense labor that we know from the HIV/AIDS early years of caregivers, of doctors, of nurses, of activists, of there's a list of people who actually kept people alive, right, when there were no therapies. And I think this has also happened with COVID. It's too early to be even writing about COVID. We will know in the decade to come what has happened, right? So that's why I think of, just the idea of X number of population, 67% of the world's population, right, are living with these antiretrovirals. If I give you that figure, right, what does that mean? It says that usually the story is the WHO, the MSF, PrEPFAR, all these big global institutions have really implemented health imperatives well, right, in all parts of the world, including the global south. But it is really HIV/AIDS knowledge, practice, and activism that has showed us that that labor of staying undetectable was distributed across protagonists. So high and low. So I think the high and low comes up as a kind of a knowledge economy organization where value accrues somewhere. And, okay, so you're given the computers. I was working in Manipur, and it was very interesting that at one point in the early years of the epidemic there was an anonymous donor who donated a PCR machine, right? And chose to remain anonymous because they didn't want the labor of these groups that had mobilized, right, to be invisible. And so then it becomes against the beneficiary argument, right? So I think the massive distributed clinical labor that I'm talking about draws attention to the fact that the project of staying undetectable is actually won at the ground level, including people who are taking their medicines. Yeah, and not at some other sort of conduit where value is produced. So that's sort of the simple answer. The complicated answer, and I really like your question, I have seen only 2 studies where the Humsafar file data have been used, one by a postdoc called Crispina, who's shared his work, basically talking, but he was really looking at who were the people who actually were non-adherent and what were the reasons and what was the success of this program. Right. So one of the ways in which the data so far has been used is registered with NACO, right, the National AIDS Program, has been used is to assess whether or not they should get the free drugs and therapies. How successful are they? Now, selling them to of course, it can always happen, and I'm not sure whether that has happened at that level. I think it's a very important point because I think in trying to stress the structural One Health program, I have recessed some of the global bioeconomy argument. And I think that's where you're pushing me towards. So thank you. I don't have a clear answer for that.
Yeah, but I think Bishnu, you have given us a kind of opening within which to think about Junaid's question, especially now because we are at the precipice of the same arrangements being rewritten under COVID. I have another covert question in chat. I'm not sure why people are not fronting, but I'm happy to read it. It says, can you explain your intriguing title, Viral Touch?
Okay, well, thank you for that question. And as titles go, they're written in blood, so to speak, right? There could be many, many, many titles. And I know my partner initially was like, why are you calling it the virus touch? A touch is so, I mean, it's got a kind of a loosey-goosey, it can be a bit too precious. Kind of idea. But then I was really interested in communicability, contact, and now with COVID-19, it seems that we are losing all kinds of touch, right? With the, not necessarily so, with the epidemic technologies like masks, right? And so I think one of the emphases in the book is to think about sort of not this kind of technical media and the kind of the calculus of health. And I showed you one aspect of it, but to think about a kind of a more sensuous engagement. And I've been using Karen Barad's work here, to think about the materiality of the scientific practices and the fact that the technologies that you use, etc., are in some ways you're caught in this flux of participation. That's the term I've been using. So the virus touch comes out of an engagement with David Abrams' work, the sense, what is it called now? The sensuous medium, I think. I'm now, I can look it up. Anyway, David Abrams, who basically is an anthropologist who was looking at shamans and people who sort of modulate your perception so that you have a sense of entanglement with the larger human world, like with the mountain or the trees, etc., right? And this he called a certain kind of sensuousness that comes about when you're caught in the flux of participation. So in the hurry to make, get the test done, in the hurry to get antigen test, in the hurry to make different vaccines, there's a kind of an urgency and a difficulty that scientists themselves encounter in the limitations of their technologies and the limitation of techniques, in the impatience that we all have, right? We keep wanting this thing fixed. Well, HIV went on for 4 decades. I'm not saying this one will, but I'm just saying that we've been remarkably impatient about this. And the scientists are caught in this flux of participation, and they're always aware of this, a sense of something else. And that's the sensuousness that touch captured. It's kind of a long answer, and you can see I'm avoiding going to COVID, but you can ask me questions about it if you like, because I realize that's the next.
I think it's a generatively queer answer, so we shall leave it at that. But we have time for one more question, and I want to throw it to my comrade in the Bay, Maitri. Take it away.
Thank you so much for this talk. And, I was thinking about it because I was, looking at on one hand, these archival sources in Sri Lanka around a detention camp in Tuticorin, and thinking about how the plantation workers were held and the types of data that was available at the time to distinguish who was the virus was transmissible and so forth. And I don't think it was cholera at the time, but that I'm also looking at other more filmic, cinematic portrayals of cholera outbreaks in the plantations, and then also the burning of line rooms and so forth. And what's interesting is, in the start of kind of these times when viruses are not detectable or you can't prove immunity. And thinking of Catherine Olivarius's work on immunocapital and this idea that you can perform or you can imagine what immunity would look like based on, or you could perform your acclimation. It's interesting to think of kind of how, you know, what was the media's role in censoring? Because we know during yellow fever in the antebellum South and also in the plantations, there was a certain element of performativity to say, all is good here, let capital flow, let, you know, let our economic, you know, our bundles of capital keep. And then there was a very much a reality that populations were just dying out. So the kind of, how was the media balancing the oral testimonies, the photographic witnessing? Definitely not for COVID, maybe you could answer, but for, you know, as we think about HIV, which has a long history of documentation in the visual aesthetic paradigms, it's interesting. So it just made me think of that and would love to hear your thoughts. Yeah.
No, a fantastic question. Also, I think because I think the part that I didn't touch on today is the biopolitical, you know, and there's the effort to constantly securitize. And the early example, of course, is the yellow fever camp, you know, Walter Reed's camp in 1901, which, you know, is also basically, you know, Cuban volunteers were given, you know, a very small sum, and they weren't actually told about risk from being bitten by infected mosquitoes. So this kind of biopolitics that make medicine possible, right, has been around for a very long time. And certainly the camp is a figure for that in many pandemic histories. I've written about that in the AIDS and the Distribution of Crisis. I think most pandemics, you know, why I think of distribution is we see very starkly, and I will talk about COVID here, I've actually written about this, is we see a population culling. Yeah, the nurses who worked in the yellow fever wards in the 1900s, and Clara Maas, you know, a particular nurse who has some fame, who basically was supporting her 5 brothers and her mother, died of yellow fever because she volunteered as an experiment, right? So there are nurses and volunteers who've always died, but that, and the Walter Reed camp, very important because that's where the consent forms get drafted for the first time. Now, if we move to COVID-19, what did it mean in March 25th to 2020 to have a national lockdown in India, right? If that, it was about the middle class who had the home to be locked down in. And I've read the million migrant march back to the villages as really, and this will sound harsh, but a population culling. And I think in the camps have really are exemplars of population culling. And it happens across, there are people who are going to die. Right. And so it was a kind of a culling, and culling, as you know, comes from the history of, you know, weakened animals on farms. Right. And I use that purposely to bring into the equation all the ways in which this human animal spectrum, which is a part of colonial racism, gets replayed again and again and again. Yeah, and in the US, of course, again, I don't need to tell anybody about the macabre Reagan politics of letting die, right? If that was not a camp, I don't know what that was, you know, in the early part of the AIDS epidemic. So I think it's unfortunately a kind of the camp is an exemplar of a general principle of distributive health in pandemics that routinely cull populations. I mean, that's sort of the sad and harsh thing that I would say in answer to your question, if that makes sense, Maitri.
Yeah. Thank you so much. Yeah.
Yes, unfortunately, as somber as that is, I think it certainly does. And I can tell you, as being in Mumbai for the last month or so, the culling of caste and Muslim subjects within this kind of unrolling metaphor of the pandemic is more abundantly clear than ever. So I think, you know, we see this not just in South Asia, but obviously in the US as well. I want to thank Bishnu for that extraordinarily generative talk. And I can see by the many comments that people are pasting that you've given them a tremendous amount to think about. And if you have not had the pleasure of encountering Bishnu's work, both previous and present, I encourage you to rush out and read it and learn from it. I also want to remind you that we have a wonderful talk coming up in a month, Meena Kandaswamy, who will be swinging her poetic wisdom and returning us to many of these questions in a different tenor. So I invite you to come back and share these conversations with us. Let's all thank Bishnu. If we were here, we would be in touch with you literally. But since we're not, if we could just give her a big hand for sharing her new work. And thank you everybody for joining us. And I look forward to seeing you again. Jai Bhim.
Thanks for coming. Bye. Thank you very much.